So, how do natural killer cell derived exosomes work to target cancer in zombie cells? I mean, this probably seems obvious, but why is this approach considered so groundbreaking?
Well, I'm going to take those in reverse if that's okay. It's
That's fine.
groundbreaking because it was so hard to accumulate the natural killer cell exosomes in the first place. And giving natural killer cells has challenges because, again, you can reject them. And to get your own requires a lot of lab work. You have to harvest your white cells, separate out the natural killer cells, which is very labor intensive, and then grow them in a petri dish so they expand. And you have to encourage them to do that and then give them back. There's a lot of work involved. The exosomes are an off-the-shelf therapy from the natural killer cells. And then how do they work? Their exosomes contain two enzymes that are very particular, perforin and I think granzyme. And if I'm wrong, someone will correct me here in the show notes or comments. But perforin actually helps perforate or poke holes in the bad cell so it can rupture. We call that lyse. It can lyse open and it can be destroyed that way. Meanwhile, they do not affect these NK or natural killer cell exosomes do not hurt or affect our normal cells. So they are very specific. They're like a cruise missile. They go right to the target.
They are such a cool cell. It's like one of my favorite structures or concepts in cellular biology and exercise physiology. So I'm going to be geeking out through this whole conversation. So
Well, I've
just
got
be
plenty of
aware.
time. We could go for hours.
And I'm sure you have plenty of information too. So when we talk about activated natural killer cell exosome therapy, how is that different? And I mean, I think the differences are probably a little bit more obvious, but how is it potentially safer than chemotherapy and radiation, which are the more traditional treatments for cancer?
Well, in answering this, I'm not necessarily saying that chemotherapy and radiation are wrong, but they certainly have their downsides. Both of them are poisonous to our cells. That's the strategy by which they work. The goal is to poison the cancer faster than we kill ourselves, so or kill our cell populations. Whereas the natural killer cell exosomes don't have any of that downside. We've never seen a side effect from using them and none have been reported in the literature. So far, they seem to be apparently safer. I'm not allowed to make that claim yet because we're not allowed to make marketing claims about safety and efficacy. But anecdotally, it looks like we haven't had any safety issues so far.
And it would seem like it'd be hypothetically, if I have to say it that way, more comfortable for the cancer patient than
Oh
going
yeah.
through chemotherapy, where
Oh
you're not losing hair, you're not
right.
getting nauseous and vomiting and all that kind of stuff.
Right. Yeah. Those side effects are common with most chemotherapy and certainly radiation and tissue damage and that kind of thing. So we're using them to help address cancer for that reason. We're also using them for people that are high risk for cancer who want to take action, do something. So maybe you have a BRCA gene mutation and your family history is positive for cancer and you want to do something. And then we have people who've already been treated for cancer in a traditional way and they're in remission and they're looking at statistics saying that they've got a good chance of being in remission for 5 years, but they want to push that to 10 and 15 and 20 years and we want to push it for them. So these might be strategies to try. And again, we can't make any claims, but I can show you literature. I can show individual patients literature, medical publications that have shown benefits.